Hello friends, this video on Kingdom Planty Part 25 is brought to you by examfear.com. No more fear from exam. So with this, I reach towards the end of this lesson on plant kingdom. So let us quickly look at a few questions just to ensure if you have understood the lesson well or not. So the first question here. What is the basis of classification of algae? So you saw that we further classified the kingdom algae. So why not the kingdom algae? In the plant kingdom, we had a group called algae under thallophytes. So we again divided algae into three types. Why did we do so? And what was the basis of that classification? The basis was the pigments presence. So the basis was the presence and absence of pigments, food storage and cell wall. So primarily this was the basis. Now, due to the presence of different types of pigments, the color of the algae was also different. So we divided them into three types, that is chlorophyce, phaeophyce and rhodophyce. Chlorophyce had chlorophyll A, B, that is why they were green in color. Phaeophyce had chlorophyll A, C and fucoxanthin, because of which they were brown in color, and rhodophyce had chlorophyll A, B and phycoerythrin, because of which they were red in color. Talking about food storage in chlorophyce, food is stored in the form of starch. In phaeophyce, it is in the form of complex carbohydrates like laminarin, and in rhodophyce, it is again in the form of starch. Talking about cell wall, in chlorophyce, it is made up of cellulose. In phaeophyce, it is made up of cellulose and algin, and in rhodophyce, it is made up of cellulose and pectin. So this was the basis of classification of algae into three types. Let us look at the next one. When and where does reduction division take place in the life cycle of a liverwort, a moss, a fern, a gymnosperm and an angiosperm? So now you have to quickly recall the life cycle of each of these plant types. And then we have to identify where reduction division take place. Now what is reduction division? Reduction division is nothing but meiosis. Now how do we know when meiosis take place? Now whenever meiosis take place, what will happen is the diploid cell will get converted into haploid. That means diploid cell will reduce to haploid cells. So diploid cells are those with two sets of chromosomes and haploid cells are those with single set of chromosomes. For example, I'll give you one example. The zygote. Zygote is a diploid. Now this zygote forms the sporophyte and then sporophyte gives the haploid spores. So this is meiosis. So you have to tell in the life cycle of each of these plant types where exactly the reduction division take place. So let us first talk about a liverwort. Now a liverwort and a moss, they both fall under the category of bryophytes. So let us talk about bryophytes first. So in bryophytes, if you see, where do you think this happens? That is, zygote forms the sporophyte and then this sporophyte gives rise to spores. So zygote was diploid, sporophyte was again diploid, but the spores were haploid. So this was the place where reduction division actually occurred. So in the life cycle of a moss, where do you think is the sporophyte? This is where the sporophyte forms. Now this is the capsule of the sporophyte and the capsule of the sporophyte contains the spores. So inside the capsule, the reduction division take place and the spores are released. So the similar is the scenario for a liverwort. Now when I talk about a fern, it is a pteridophyte. So in case of a fern, if you see, these are the spores. Where are the spores present? They are present in the sporangia. So here if you see, meiosis occurs. So when the spores are released from the sporangium. So here exactly the meiosis or the reduction division take place. So next if you look at the angiosperms. So in the angiosperm, the anther of the stamen will have the pollen grains and the ovary of the pistil will have the eggs. So where exactly the meiosis will take place? So here if you see, this is the place where the gametes are produced and then the gametes fertilize to form the zygote. And this zygote will form the sporophyte and then the sporophyte will again release the spores. So this is the place where exactly the reduction division take place. That means the reduction division will take place in the anther and in the ovary of the pistil, because that is the place where the spores are actually formed, or the gametes are actually formed. So if you remember the life cycle, you can actually tell where exactly what happens. Let us look at the next one. Name 3 groups of plants that bear archegonia. Briefly describe the life cycle of any one of them. So what is archegonia? Archegonia is nothing but the female sex organ that produces the female gametes. So archegonia was a kind of another name for the female gametophyte. So it was that sex organ which produced the female sex cells. So plants bearing archegonia were bryophytes, pteridophytes and gymnosperms. Now, if we want to describe the life cycle of any one of them, it is again the same thing. Let us suppose if we describe the life cycle of a pteridophyte like fern. So this is how the life cycle would be. The mature sporophyte is the dominant phase. This will form in the sporophyte. If you see, it has the sporangium. These leaf-like structures are called sporophylls, and on the sporophylls you have small structures called sporangium. Inside the sporangium are present spores. The spores are released. The spores will then germinate to form a gametophyte. This gametophyte then will have the male and the female gametophytes, which will form the male and the female sex cells. They will fuse together to form a zygote. Then the zygote will grow into a new sporophyte, and that will again in turn form the mature sporophyte. So this is the life cycle of a pteridophyte. Okay. Let's look at the next one. Mention the ploidy of the following. What do you mean by ploidy? That means whether it is a haploid or a diploid or a triploid. So we have to mention how many set of chromosomes are present in each of these cells. Protonemal cell of a moss. Protonema. Which was protonema? Protonema was the stage which was formed before a gametophyte. Remember I was telling? Okay. So this is haploid. Primary endosperm nucleus in a dicot. So when is a primary endosperm nucleus formed? Primary endosperm nucleus is formed by the fusion of a male gamete and the diploid secondary nucleus. So this male gamete is haploid and this is diploid. So haploid plus diploid will form triploid. Therefore, primary endosperm nucleus is triploid. Leaf cell of a moss is again haploid. Prothallus of a fern, prothallus, it is again haploid. Gemma cell in Mercantia is again haploid. Ovum of a liverwort, ovum is nothing but the female gamete, so it has to be haploid. Zygote is diploid because zygote is formed by the fusion of a haploid male gamete and a haploid female gamete. So haploid plus haploid will form a diploid. See, all these haploid, diploid will become more clear when you actually go through the life cycle of each plant. Next is, what is heterospory? Briefly comment on its significance. Give 2 examples. Heterospory. Hetero means different, spory means spore. So it is the phenomenon in which the plant produce different types of spores. That means all the spores which are produced by the plants are not similar. 2 types of spores are produced, megaspores and microspores. Megaspores are bigger in size and microspores are smaller in size. So the bigger megaspores give rise to the female gametophyte and the smaller microspores give rise to the male gametophyte. Now talking about its significance, these different types of spores ensures protection of the gametophyte. It also ensures that the development of the zygote takes place inside the female gametophyte. So when the development takes place inside the female gametophyte, so the zygote will remain well protected. Next is match the following. So we are given 2 columns and we have to match these examples of plants with their corresponding groups. Chlamydomonas is an algae. So here is Chlamydomonas. Cycas. What is Cycas? Cycas is a gymnosperm. Selaginella. Selaginella is a pteridophyte. And the last one is sphagnum, which is a moss. The sphagnum is the one which acts as a very good packing material. Okay. Again, let us look at some distinction between red algae and brown algae. So many important differences. Red algae are known as rhodophyceae, brown algae phaeophyceae. Why are they red? Due to the presence of chlorophyll A, B and phycoerythrin. Phycoerythrin is in greater amounts. Brown algae is brown due to chlorophyll A, C and fucoxanthin. It is basically due to fucoxanthin. Food stored in red algae is in the form of starch, whereas in case of brown algae it is in the form of complex carbohydrate like laminarin. The cell wall of red algae is composed of cellulose and pectin, whereas in brown algae it is made up of cellulose and algin. So these are some of the differences between red algae and brown algae. Distinguish between homosporous and heterosporous pteridophyte. Just now I was talking about heterospory, right? Homosporous, homo means same, hetero means different. So heterosporous spores which are produced are all of same type and size. In heterosporous, they are of different size and types. So when I talk about homosporous, the bisexual gametophyte is produced because all the spores are of the same type. So when those spores will actually give rise to the gametophyte, it will be bisexual. That means both the sexes will be present in the same gametophyte. Whereas in case of heterosporous, unisexual gametophyte is produced. Example of heterosporous is most pteridophytes, for example, equisetum. Example of heterosporous would be some of the pteridophytes, not all. Example is Selaginella. Let us look at the last to distinguish between syngamy and triple fusion. What is syngamy? Syngamy is a name given to the fusion of a male gamete and egg. You remember we spoke about double fertilization which take place in angiosperms. What is double fertilization? There are two fusions which take place inside the ovary. What are they? One is the fusion between the male gamete and the egg, and the other one is the fusion of the other male gamete with the polar nuclei or the secondary nucleus. Right? So this fusion of the male gamete and the egg is known as syngamy, and the fusion of the polar nuclei with the male gamete is known as triple fusion. So what is the result of syngamy? Zygote is formed which later develops into an embryo, and the result of triple fusion is endosperm is formed. So now then the zygote starts developing as an embryo, and this endosperm keeps providing nourishment to that developing embryo. So this is the difference between syngamy and triple fusion. So now we have reached towards the end of this lesson. I hope that this lesson on plant kingdom helped you to understand the different categories of plants and how can we understand the life cycle of each of them. Please focus on the life cycle of each plant type because that is the most important thing in this lesson. So hope this lesson helped. See you all in the next lesson. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.